Welcome, ladies and gentlemen, to the Zhittya Genesis Medicine webinar on Parkinson's disease. Is therapeutic angiogenesis a treatment or even a possible cure? I'm Daniel Montano. I'm the Chief Executive Officer of Zhittya Genesis Medicine. We have over 525 people that have registered for this today, and we hope that you will enjoy it. Zhittya is a biotechnology company that is developing a biological pharmaceutical that we believe can address the pain and suffering and cause of death for the diseases that kill over 50% of all adults on Earth. Heart disease kills 38% of all adults. Stroke kills 12%. That's 50%, and there's still 73 additional cardiovascular drugs that cause suffering and death. Over the last 20-some years, Dr. Jacobs and I have expended over $140 million advancing our medical treatment to treat the diseases characterized by lack or insufficient blood flow to a tissue or organ. Zhittya's business is basically two things. Number one is discovery of pathways, discovery of dosing, discovery of what our drugs can do, how we can use angiogenesis to treat Parkinson's or Alzheimer's or heart disease or peripheral artery disease. And after the discovery, then number two, our business is to produce the clinical evidence to support the government regulatory approval requirements of a pharmaceutical drug. To provide you some evidence of our prior success, I'm going to show you a short ABC nightly news clip on our drug being injected into the human heart of a no-option heart patient. This is a woman who her doctors had told her she had less than a year to live. She volunteered for our clinical trial at the University of Cincinnati, which was cleared by the US FDA. She volunteered not because she wanted to be an experimental person. She volunteered because she was told she had 1 year to live. You will be able to see the angiogram of her heart before our drug was injected. You will see how plain it is and a lack of blood flows. Subsequently, again, you will see an angiogram after our drug is injected into her heart, and you will see a new field, a new area of vessels providing sufficient blood to her heart muscle to allow her to limit the pain in her heart and allow her heart to function again. Now, Dr. Jacobs will be going into a lot of this, but I want to emphasize something. When it comes to the brain, we do not know if our molecule will work there in humans. Jack will talk about animal data. We're in the process of doing the human brain analysis now. But when you look at this ABC news clip, look and see what it did in the heart muscle and how it provided blood in the heart muscle. If it can do the same thing in the brain, we believe we can enhance the recovery from strokes. We believe we can reverse Parkinson's, ALS, Alzheimer's, chronic depression, and more. I'll show you the video now.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
Now, Dr. Jacobs is going to be giving a presentation, but just for your additional information, if you wish additional information, we have a small booklet that talks about the hypothesis of angiogenesis providing new blood flows to the heart muscle to treat heart disease, to the brain to treat stroke and others. It goes to the hypothesis, and it will then also have 2 human clinical data pools, one on diabetic foot ulcers, one on the heart, and we'll also have the data on Parkinson's disease in animals. Additionally, after the presentation, if you want more information on Parkinson's disease, Dr. Jacobs has written a white paper that provides the animal data and what we're doing, etc. If you want those, you can email me at dan@zhittyamedicine.com, and that will pop up on the screen every now and then, and I will send you the white papers. After Dr. Jacobs' presentation on Parkinson's disease, we will have a 10-minute question and answer period. You can send your questions by clicking on the Q&A button on your Zoom screen. Drug discovery is a slow, difficult, and risky enterprise. There are no assurances of our success. Please be part of our discovery is learning what will not work. We thank you for your time. And additionally, now I get to introduce Dr. Jacobs. Dr. Jack Jacobs has a PhD from Washington University, St. Louis, Missouri in molecular biology. Jack has worked at Merck Pharmaceutical, Hitachi Pharmaceutical. Jack, besides being one of the smartest people I have ever met, is also one of the nicest people I've ever met. I'd like to introduce now Dr. Jack Jacobs, my partner for the last 25 years. Jack?
Thank you, Dan, for that kind introduction. So thank you for viewing in with us. I know it's late for some of you if you're viewing from Europe. I'm going to also be giving this talk on Parkinson's disease at the end of September 29th. So if you happen to nod off halfway through my talk, you can catch the second half at the end of September. Now, before I start talking about how we think therapeutic angiogenesis may be a treatment for Parkinson's disease, I just want to update where we're at on our clinical trials for Parkinson's. I know a lot of you have been following us closely, and some of you already probably heard most of this talk, but I want to give you all an update on our clinical trial status. Okay, so in the United States, as you know, we've submitted an investigational new drug application, an IND application, to treat subjects with Parkinson's disease. This was reviewed by the FDA, and they have requested an additional animal study. As this was the first clinical trial proposed to study our drug, FGF1, in a brain indication, they wanted some additional toxicity study. Took us about 3 months to go back and forth with the agency on what type of toxicity study they wanted, and we've engaged an animal lab to do the study, and we actually first sent it to the FDA and they accepted it, and we're now undertaking that study. It's going to be about a 9 to 12 month study. Now, at the same time, we submitted the same regulatory packages to countries outside the United States. In Mexico, for example, we submitted our Parkinson's protocol, and in fact, they felt that the protocol was fine, that our animal data was fine, that we did not need additional animal work, and we've gotten approval to begin that study in Mexico. We'll be giving the drug 5 times a week for 4 weeks, and I'll talk more about the clinical trial at the end of the talk. We've identified a very talented neurologist in Monterrey, Mexico, Dr. Martinez, and his hospital, Hospital Zambrano, is probably one of the premier hospitals in all of Latin America. It's modern and it's superbly equipped. Now, in April, we were informed because of the pandemic that all clinical trials except for the COVID-19 clinical trials will be on hold until further notice. Our clinical research organization in Monterrey has informed us that perhaps in the October-November timeframe, we may be able to get going. In addition, we've submitted a separate independent clinical trial for patients with ALS or Lou Gehrig's disease, and that also has been approved to begin in Monterrey. And in the coming months, we also hope to submit additional indications in Mexico for Alzheimer's disease, for the heart indication, and also for stroke recovery. We also submitted the Parkinson's disease protocol to the Baltic country, Estonia. We've been informed Estonia is now allowing clinical trials to proceed. We've identified a principal investigator there, Dr. Tumsu, who's a neurologist and who practices in the capital of Estonia, Tallinn, at the East Tallinn Central Hospital. We have a final application which is now going to be considered by the Estonian FDA, which is part of the European Union. Again, it will be the same study as will be done in Mexico with 3 different doses of the drug being studied. So we hope to have 2 independent clinical trials going with Parkinson's disease that allow us to compare and contrast the results that we're getting. Okay, I'd like to start my talk now, and as Dan mentioned, we're very interested in blood perfusion, growing new blood vessels, and in fact, over 75 human diseases are a result of impaired blood perfusion to a tissue or an organ. Some of those are very obvious. If you block your coronary arteries, you're going to have problems in your heart. If you block an artery going into your brain, you're going to suffer a stroke. But over the last 5 to 10 years, a number of medical research groups have also shown that lack of blood perfusion initiates some of the neurodegenerative diseases such as Alzheimer's, Parkinson's disease, and ALS. So we'll be talking about those in future webinars, but today I'll be talking about Parkinson's disease. Now, before I get into that, I'd like just to show you a study which was published recently from a very prestigious medical school, Columbia University Medical School in New York. This was published in the peer-reviewed journal Cell, and it really supports our hypothesis that a lack of blood flow to the brain can cause problems with diseases. What they showed, and what you'll see in a short video that was made from this study, is that even as we age, as our brains get aged, a lack of blood flow contributes to the problems of aging. So let me show you this video now. It's a short clip, again, from Columbia University Medical School study. Why our brains perform worse as we age. So the researchers there studied 28 healthy individuals who had died suddenly from accidents. The people ranged in age from 14 years to 79 years of age. So they looked at the number of, mainly it's stem cells they were looking at the study, stem cells in different parts of the brain. They found that older people made as many of these stem cells in the memory parts of the brain as younger ones do. But this is critical. What is different in the aging brain is the reduced blood flow to nourish these stem cells. So in older brains, this means the stem cells are dividing less than they are in the younger brains. So the pool of stem cells is still there in the older brains, but just not as active. So the researchers at Columbia University suggested that it may be possible to combat age-related cognitive decline if we're able to improve blood flow to the brain. So we could not agree more with the findings of this study. This was with healthy individuals. We believe the same thing is going on in different parts of the brain in these various neurodegenerative diseases. Now, let me show you the results of another study using relatively newer imaging technique called functional MRI. Okay, and this looks at regional blood perfusion. So you can look at very small areas in the brain, look at blood flow in those areas. This is a very accurate and sensitive technique. And what the study did was to look at healthy 21-year-olds in blue here. They have maximal blood flow in their brains, and they were focusing in on the area of the brain that's affected in Parkinson's disease. So this is deep in the midbrain where the dopamine containing cells are. So that region of the brain, healthy 21-year-old maximal blood flow. As we saw in that video, as we age, this is a healthy 65-year-old, we get reduced blood flow throughout the brain, including where the dopamine cells are. But the dopamine cells are not dysfunctional at this level of perfusion. However, when we see Parkinson's disease patients, we see almost a 50% decrease in blood perfusion in the area of the brain where the dopamine producing cells are. We think that leads to dysfunctional dopamine producing cells and causes the motor symptoms of Parkinson's disease. People who've had a stroke in that region of the brain have almost a complete cessation of blood perfusion in that area of the brain. So how could we perhaps get someone with Parkinson's disease up to where this healthy 65-year-old is? How can we increase blood perfusion in a person with Parkinson's disease? Well, one obvious way is to get more blood vessels grown in the brain, which would bring more blood perfusion. And so that's what we're going to be talking about. We're going to be talking about growing new blood vessels, a process referred to as angiogenesis. And we'll be talking about a drug, our drug, FGF1, which is probably the most potent stimulator of angiogenesis, or new blood vessel growth in the human body. Okay, we use angiogenesis all the time. It's a natural process. You use it whenever you cut yourself, say. This is an example of angiogenesis during wounding. Think of this dotted circle as a scab. We're looking underneath that scab, a wounded area here. These little tendrils of new blood vessels, that's angiogenesis being stimulated by our drug, our growth factor. This angiogenesis then stimulates the regeneration of skin tissue. And all of skin, not just the skin cells, but it regenerates skin as an organ. We have regeneration of nerve cells in there, hair follicles, sweat glands. And so it kicks off that whole wound healing process. Now we've taken the drug, FGF1, and are now using it in these clinical trials in what we call therapeutic angiogenesis, okay? So think about this. This is, Dan showed you that video of the heart patient. Think of this as a person with heart disease, stunted blood vessels. These are coronary arteries. And as we saw in that video, we inject the drug, our drug, FGF1, directly into the heart muscle. And 3 months later, we get this robust blooming of new blood vessels, which allows the patients to function better. And I'll be talking about this coronary artery disease indication in about a week at a separate webinar. We'll talk now a little bit about our drug, its mechanism, and then I'll get on into the work on Parkinson's disease. Our drug is FGF1, fibroblast growth factor 1. It's a natural growth factor present in our bodies. I just showed you how it works with wound healing. It's most abundant in our brains. In fact, many years ago, I was involved in purifying it from cow brains. I'll talk about at the end of this talk how we use genetic engineering, bioengineering to make our drug for the clinical trials. It's a potent stimulator of angiogenesis. So let me talk a little bit about angiogenesis now. This is a slide we borrowed from the Angiogenesis Foundation in Boston. Here are the angiogenesis factor. This would be FGF1. And the key initiating event in this whole process is when FGF1 binds to this little Y-shaped receptor, okay? And that's sitting on endothelial cells, which are the cells that line all of our blood vessels. So we get this binding, endothelial cell receptor binding, and that kicks off a whole explosion of activities resulting in the endothelial cells beginning to divide, and they naturally form these little tubules. Okay, this is the beginning of a new blood vessel. And over time it extends. Here's a capillary. And then this process is repeated thousands of times along this vessel here. Now what's unique about our drug, FGF1, is that it also could stimulate small arteries called arterioles. We see that here. So basically, an arteriole is a small capillary which is surrounded by these smooth muscle cells. And that gives these vessels a bit of a pumping action, so you actually get better perfusion with these small arterioles. All right, let's look at this interaction of our drug with the receptor, because this mechanism is very important to understanding how our drug works as a therapeutic. Okay, so here's our FGF1 drug coming down to its receptor, this Y-shaped receptor, on the surface of the endothelial cells, which are lining the blood vessels. That causes an explosion of metabolic events. I'm not going to go into all these. There's thousands of papers that have been published on all these pathways. But the net result is the endothelial cells start dividing, and you get those tubules formed, and then new blood vessels form from that. Now, I'm often asked why, when we give our drug to people, why don't we see blood vessels growing throughout the body? That's a very good and important question. Obviously, we don't want blood vessels growing in the eye or in the retina. They could cause blindness, clogging up the kidneys. So why does our drug only work on where it's needed in damaged tissue? So let me just give you that mechanism. This is critical to understanding how our drug works in the body. So let's use a heart disease as an example. So a healthy heart becomes diseased over time if someone develops heart disease, coronary artery disease, over time. And what happens over time is that tissue becomes ischemic or starved for blood, oxygen, nutrients. And work from a number of labs have looked at the FGF receptors in the heart and what happens over time as a person develops heart disease. So in the healthy heart, there are relatively few FGF1 receptors, mainly because the healthy heart doesn't need additional blood vessels. But as the heart becomes diseased, and we reach this state where it's ischemic, where it's lacking blood, the endothelial cells are basically crying out to start dividing, to form new blood vessels, to get more blood into the heart muscle. And there's an upregulation here of the FGF1 receptors as shown here. And in fact, it's known that you have to have about 10,000 of these receptors occupied with the FGF1 drug to get that explosion of metabolic events that lead to new blood vessel formation. So healthy heart, there might be 100 receptors, not much is happening. In the unhealthy heart, it's primed to react to FGF1, but for whatever reasons, there's not enough FGF1 around. And that's where we come in with our therapeutic angiogenesis. We actually inject the FGF1 right into this situation. And an artist's rendition is shown here. So here's the starving heart, the endothelial cells upregulated, their FGF1 receptors. We come in with our drug, FGF1. Therapeutic angiogenesis is initiated here, and we get new blood vessels formed in the heart, just as we saw in the video that Dan showed you. Okay, so we're going to use this approach in a number of diseases that are caused by a lack of blood flow to an organ or a tissue. And I'll talk about these indications in upcoming webinars. We'll talk about its use as a wound healing agent to heal diabetic foot ulcers. We'll be injecting it into the leg muscle to help with peripheral artery disease. In a week's time, I'll talk about its use in coronary artery disease. We'll talk about stroke. And today I'll be talking about Parkinson's disease, but in upcoming webinars, we'll talk about its use in Alzheimer's disease. Vika Montana will be talking about its use in multiple sclerosis. And so please tune into those upcoming webinars as well. Okay, your brain is incredibly vascularized, probably the most vascularized tissue in your body. There are billions of capillaries in the brain that feed and nourish billions of neurons. FGF1 mainly stimulates the microvasculature, capillaries, and small arterioles. So that's what we're going to be focusing in on this talk. And a new term, the Neurovascular Unit, has become very popular. This is the interplay between neurons in the brain and blood vessels in the brain. So go from the whole brain, the large arteries, branching down to these smaller capillaries or arterioles. So this is a capillary supplying this adjacent neuron with blood, with oxygen, with nutrients, and also very importantly, removing waste products from that neuron. So you can imagine, and not really shown here, are also the pool of stem cells which were referred to in that Columbia University video. But you can imagine if this capillary gets blocked, let's say by atherosclerosis, or genetic defects can lead to blockages, traumatic injury, concussions, inflammation can lead to this being blocked. So any number of events can cause these capillaries to be blocked, leading to damage of the neurons in that area. And again, as that video showed you, stem cells also need adequate blood perfusion to divide and mature. So these guys are pretty much backup neurons. As neurons get damaged in the healthy brain, these then differentiate to form neurons. So for example, in the dopamine containing cells, these stem cells would give rise to new dopamine cells, as well as other supporting neuronal cells, which are all needed for healthy brain tissue. So again, lack of blood flow not only can affect the existing neurons, the existing dopamine neurons, but also the pool of stem cells, which are needed to replenish neurons as they become dysfunctional. And this just shows you, this is in a rat brain, shows you what normal vessels look like in a brain. This is a very dense network of blood vessels. These are capillaries. Not shown here are the neurons, which would be fed by all these capillaries. But look at what happens when we give that animal an experimental stroke. We decimate the capillary beds, as shown here. So one day later, after giving this animal a stroke, we decimate the capillaries. We give one of the sets of animals FGF1, and you can see here, 14 days later, we have this nice new network of capillaries. Not quite back to what this is like, but still much better than without drug. This is without FGF1. The brain desperately tries to reestablish perfusion, blood vessels in the damaged area, but it really can't do that well of a job. You can see tangled, dysfunctional blood vessels. These actually can hemorrhage and cause further neuronal damage. So the animals here are not doing nearly as well as these animals that have been given FGF1. And I also should say that this drug was delivered intravenously, so we know that FGF1, at least in these animals, is crossing the blood-brain barrier. Okay, so let's talk now about Parkinson's disease. I'll talk a bit about some of the animal studies. I'll end up giving some details on the clinical study that we'll be doing. And also want to talk a little bit about the drug that we'll be using, how it's manufactured for the clinical trials. Okay, so as most of you know, Parkinson's disease affects dopamine producing neurons. There's only about 500,000 of these neurons in our brains, and they're located very deep in the midbrain, right in this area. That's called the substantia nigra region. And we believe, due to a lack of blood flow in that region, that these cells become dysfunctional. So normal neurons with normal movement become dysfunctional in Parkinson's. Less dopamine is secreted, leading to the motor disorders seen in that disease. And in fact, many of the medications which are given to Parkinson's patients replenish that dopamine and work for a period of time, but eventually they stop working. We also know that in Parkinson's, other areas of the brain can be affected. For example, we know that smell is one of the first, dysfunction of smell is one of the first symptoms seen in Parkinson's disease. And what I'm going to show you here is, we're going to look at the area of the brain involved in cognition. This is a woman who has Parkinson's disease, but also dementia. And we're using that same colorized MRI functional scan to look at her brain. Blue is normal blood flow. Yellow is less blood flow, and then red here is severely reduced blood flow in this woman that has dementia. So if we gave this person FGF1, we would hope not only to increase perfusion where the dopamine containing cells are deep in her brain, but also in these areas in her frontal cortex where memory function is located. So we would hope to also improve her dementia with that treatment of FGF1. Okay, let's look at some of the animal studies that supported our regulatory submissions. There are basically two models of Parkinson's disease in animals, a rat model and then a primate model. So you can give this guy Parkinson's by giving him a toxin which selectively destroys the dopamine cells in the brains of these animals. And that's shown here. We test motor skills in these rats. There's a normal animal with normal motor skills. You give that rat this toxin, which selectively destroys the dopamine-containing cells. You can see much less a drop in the motor skill scores as shown here. When we give these animals, again, intravenously, FGF1, and again, it's getting through the blood-brain barrier. These animals improve, about twofold improvement in their motor scores. Not quite back to normal, but certainly better than the animals that did not receive FGF1. Now, importantly, if we look in the brains of these animals, so we euthanize the animals, do brain slices. And let me just say, for those of you who believe in reincarnation, you don't want to come back as a laboratory animal. But here we see in the brains of these animals, we're looking at the dopamine-producing cells that stain brown. And we can see in the substantia nigra region, the newly created, regenerated dopamine-producing cells in the brains there compared to animals that were not treated with FGF1. So this is a disease-modifying activity. We're actually replenishing the rat's brain with new dopamine-producing cells. We believe that gives rise to the better motor function in those animals. Okay, probably the gold standard for drugs that are going into the clinic for Parkinson's disease is the primate or monkey model of Parkinson's. And again, we can give this animal a toxin which selectively destroys the dopamine-containing cells in their brains. Here's that toxin. It's MPTP. It's a herbicide. It's related to paraquat. Actually, its discovery was actually tragic in the late 80s when marijuana was still illegal in the Seattle region. A marijuana grower was sprayed his marijuana crops with this herbicide. Some teenagers broke in and stole the marijuana and smoked it and came down with irreversible Parkinson's disease. So that was a real tragedy. But the same toxin, when given to the monkeys, this again is motor scores. They progressively come down with all the classical symptoms here of Parkinson's disease, the gait disorders, the tremors. We then treat one group of animals with FGF1, as shown in blue here. And we can see remarkably a halting of that progression in decline in motor skills and actually improvement in motor skills, so where they get back to about 75% of normal. Without treatment with FGF1, the monkeys continue to decline. And so when we end the experiment here, there's a great difference between the motor scores of those given FGF1 and those not. Now, I'm asked often, why didn't we keep going? Would that have gotten better? And most likely they would have improved more if we treated them longer. But at this point, we sacrificed the animals. Their brains were dissected. And I want to emphasize we're not going to be doing any brain dissections in our clinical trials, so don't worry about that. But we looked in the brains of these animals just as we did in the rats. And we looked at the dopamine-producing cells. And again, in brown here, you can see replenishing of the dopamine-producing cells in the brains of those monkeys, where without treatment, you see none of the brown staining dopamine-containing cells. So again, this is a disease-modifying activity. We're regenerating dopamine cells, and we believe that's one of the reasons these animals are doing better in terms of their motor scores. Now, also in these animals, as we see in humans, we see a toxic tangle of proteins, and this is common in neurodegenerative diseases. In Alzheimer's, you see the beta amyloid plaque accumulating. In Parkinson's, it's another protein that's called alpha-synuclein. And in both primates and in humans, this toxic tangle here, this is in the substantia nigra, the nigrostriatal region of the brain. These are actually toxic to the neurons. And these tangles get passed along down the neuronal pathway, so they almost spread and infect other neurons. You can see the animals that were given FGF1 here, less of those tangles, not quite back to the normal brain, but certainly better than this. And we believe that's a result, again, of new perfusion in the brain, being able to flush away these tangles and getting these things more back to normal. So these two activities we think are critical for the monkeys getting better, replenishment of the dopamine-containing cells, flushing away of these toxic tangles. And those two disease-modifying effects, we think, are reversing Parkinson's disease in these monkeys. This is why we're so excited and want to get into the clinic as quickly as possible to see if we see similar effects in people. So this is the design of our phase 1 clinical trial, which hopefully will start first in Mexico. We'll be looking at 3 ascending doses of the drug. So we'll look at 1x, 2x, and then 5x doses. The dosing will be done sequentially, so we'll start at the lowest dose and make sure that's safe before we go to the next higher dose. Importantly, there'll be no placebo group. All patients will receive a 1-hour intravenous infusion of FGF1 5 days a week for 4 weeks. And all the patients will have a central PIC line put in, so we don't have to stick them every day to get the drug in, or when we take our blood draws for the lab testing. We'll monitor both safety and effectiveness, and we'll follow the patients for up to a year. Now, I want to emphasize in the first clinical trial, we'll be dosing this way. We'll be using certain doses of the drug, but as long as the drug is safe in people, we can try different types of clinical studies. We can increase the doses. We can dose for longer. So this is our beginning crack at this, and as long as it's safe, we can try different techniques to eke out the effectiveness, as hopefully we saw in the monkey study. Now, I mentioned I want to talk just a little bit about the drug, FGF1, that we're using in these clinical trials. Lots of academics work on this, but we're really the only ones that have pharmaceutical grade FGF1 to do these clinical studies. So we have an advantage. And I mentioned earlier that FGF1 was first purified from cow brain, and obviously we can't put cow FGF1 into people. And in fact, FGF1 is really so in such trace quantities in our bodies, it's not even feasible to get it from people. So we've used the techniques of bioengineering, genetic engineering, and we make unlimited quantities of the FGF1 in bacteria. Okay, so this is our manufacturing plant. We grow the FGF1 up in a fermenter where the bacteria divide every 30 minutes. And as a biochemist, I just have to show you one biochemical slide. This is a protein gel where we analyze the FGF1 that's made by these bacteria. So we basically take a little clump of bacteria, bust them open, and then run these gels. Each one of these blue lines is a protein. Okay, so we put an electrical field and the proteins start migrating. The big ones stay at the top of the gel. These are all bacterial proteins. And look down here. This is the human FGF1 being made in this bacterial cell. It's the dominant protein made in the bacterial. See, it's a darker staining. So we've forced this bacteria to make a protein, doesn't really need for anything, doesn't really want, and it's the predominant protein made in the bacterial cell. Now, these bacterial proteins are toxic, so we need to get rid of them. And over a period of weeks, we purify the FGF1 till here. At the end, we have pharmaceutical grade FGF1 that's ready for the clinical study. Okay, this is my final slide where we're looking at what we hope to see in our clinical study. As I mentioned, there's about 500,000 dopamine producing cells in our brains. As we saw from the Columbia video, as we get older, we have less blood perfusion. That causes the dopamine cells to decrease in a healthy aging individual, but not to a point where they have Parkinson's disease. However, in Parkinson's, there's the inexorable decline in dopamine-producing cells to where we get down to these red dotted lines where a person has lost between 60 and 80% of their dopamine-producing cells. And that's where the symptoms of Parkinson's disease first manifest themselves. So we'll be treating patients here in our clinical trial, we hope, through the process of angiogenesis, growing new blood vessels in the brain, coupled with neurogenesis, the growing of dopamine producing cells, that we can pull these patients back up into this region where their symptoms of Parkinson's disease start to abate. So I want to thank you for your attention to the webinar. If you want to participate in the clinical trials or want more information on the company, you can go to zgm.care. This is our website. And now we'll open up the webinar to some questions and answers. Thank you.
We have been overwhelmed by questions, so I'll be asking a select few. If your questions were not answered, feel free to email dan@zhittyamedicine.com to get those questions answered directly. Our first question. Several people have asked similar questions. For these questions, I'm going to conglomerate them into one question. Is this possible treatment for Parkinson's disease something that might treat multiple systems atrophy, ALS, multiple sclerosis, and Alzheimer's?
Yep, very good question. And I will answer yes to all those. We will be studying, as I mentioned, ALS or Lou Gehrig's disease in a clinical trial in Mexico. You'll hear from Vika Montano how we can use FGF1 to treat the vascular dysfunction that we see in multiple sclerosis. Alzheimer's for sure, a very large study at McGill University points to that a lack of blood perfusion is the initiating event in Alzheimer's. So we're really excited, and we'll be able to start that trial in the coming months.
Another question was asked. How do you participate in the clinical trials for Parkinson's disease? And what would those trials cost?
Okay, as I mentioned at the end of my talk, you can go to our website, ZGM Care. We have sign-up forms for all the different medical indications that we'll be doing. You'll get on our mailing list there, and we'll be sending you updates when the clinical trials will start, which countries they'll be starting in. And we bear all the costs for the clinical trials. Even for some, I think we would consider transportation and housing. Dan might have a different view, but that's my view.
Another question. In the monkey study on Parkinson's disease, why did the improvement in the monkeys which received the drug stop at the end of the chart?
Right, so I kind of answered that in my talk. We could have treated the animals longer, but as in science, you stop the experiment at some point, and really we wanted to see what was going on inside the brains of those animals.
All right, another question. Why did Zhittya pick Mexico and Estonia to conduct the Parkinson's disease clinical trials?
We actually had met some people at the bio conference, and they told us how receptive those two countries were to studies from US biotech companies. So we gave it a shot, and we were pleasantly surprised.
All right. What are some of the side effects of taking FGF1?
Well, we've put FGF1 into hundreds of people, and we've not seen any severe adverse events that are attributable to the drug. So we monitor very closely. We do eye exams to make sure nothing's going on in the retina. We measure tumor markers to make sure nothing is going on there. But so far in our clinical use of FGF1, we've seen no serious side effects.
Another question. How will your growth factor affect cancer?
Okay, so work from a number of labs has indicated that FGF1 cannot initiate or cause cancer. But however, it is a potent growth factor. So because of that, we will not allow anyone into a clinical trial who has active cancer. So we screen people before they enter our trial just to make sure that the growth factor won't exacerbate a cancer. But I want to emphasize that it does not cause cancer when given in very, very large quantities to animals.
Another question. Does FGF1 clear the blood-brain barrier?
Yes, in animals, a number of studies have shown that FGF1 can clear the blood-brain barrier. Using radioactive FGF1, you can see it crossing and getting in. And I want to emphasize that in the animal studies, the FGF1 was given intravenously. So we're hoping that the same will be true in humans.
Another one. How do you target angiogenesis to occur in a specific area of the brain while at the same time preventing capillaries developing in unwanted areas?
Right. So I made a point of that in one of my slides where it's very important that the FGF1 be directed to the area where it's needed. So in the heart, we inject it directly into the heart. In the legs, it will go directly into the legs where they're needed. In the brain, we're giving it intravenously, so it will flood the brain, but again, it will do nothing in normal brain tissue, only in those areas that are lacking blood flow, where there's ischemia in the brain, will it stimulate new blood vessel formation.
Another question for Jack. I see that you have scheduled other webinars for the brain, enhanced stroke recovery, Alzheimer's, multiple sclerosis, and chronic depression. Are they the same as this presentation on Parkinson's disease? And would they be worthwhile for me to view?
Oh, definitely worthwhile for you to view into. Not only to see beautiful Dan Montano, but to hear about how they're all really different. For example, diabetic foot ulcers, we use it topically as a wound healing agent. For the heart and leg, we're directly injecting it into the muscle to get the muscle better perfused. And the brain, there's even different mechanisms of action. With Parkinson's, we believe it's creating new blood perfusion, stimulating new dopamine producing cells to be formed. But as Vic Montana will tell you, with multiple sclerosis, we feel it's actually repairing leaking blood vessels. And we believe the leaking blood vessels let immune cells and toxic proteins out of the bloodstream to attack the neurons that are found in the lesions that are seen in multiple sclerosis. So I think you should tune in to all of our upcoming webinars, and I guarantee they'll be different.
Well, that's unfortunately all the time we have for questions. We'll be going on to the closing statements.
Thank you, Dr. Jacobs, for your presentation. And I'd like to thank all of you who have come in and watched the webinar, our audience. I know some of you are watching from Korea, some from Kuwait, some from Germany, and many areas around the world and across the United States on how our drug may treat Parkinson's disease. We thank you for your valuable time. Our next upcoming webinar is going to be a week from now on coronary artery disease. Is therapeutic angiogenesis a breakthrough medical treatment for the #1 cause of death in the world? That'll be Tuesday, August 11th at 10 p.m. Pacific, 1 p.m. Eastern, 7 p.m. Germany, 8 p.m. Saudi Arabia. Our third presentation will be on diabetic foot ulcers, therapeutic angiogenesis as a revolutionary new treatment. If you know people with diabetes, they would be very interested in this topic, since diabetic foot ulcers affect 7 to 10% of all diabetics every year. They care about this topic. Infected diabetic foot ulcers can lead to amputation and even death. The presentation will be Thursday, August 20th, 8 a.m. Pacific, 11 a.m. Eastern, 5 p.m. Germany, 6 p.m. Saudi Arabia. Then after that, there will be a presentation on stroke recovery. Can therapeutic angiogenesis enhance the recovery from stroke? That'll occur on Monday, August 24th at 9 a.m. Pacific, noon Eastern, 6 p.m. Germany, 7 p.m. Saudi Arabia. We will attempt to host a medical webinar once a week on different medical indications. We have over 18 medical indications that we're advancing towards regulatory approval. Also, given the status of updates on certain clinical trials, as an example, what is going on with Parkinson's disease in Mexico or Estonia. Dr. Jacobs says he wants to start each webinar with that update for you. So even if you only listen the first 5 minutes, you'll get the latest news on where we are. If you heard the basic presentation on Parkinson's, it will not change much in 60 days. However, we are now reaching out to tens of thousands of people every week to learn what we're doing. Over the next 30 days, we will have an outreach campaign to over 1 million people. Remember, every week, 1,000 people in the United States discover they've been diagnosed with Parkinson's. 100,000 people a month in the world discover they've been introduced or they have Parkinson's disease. If we send out and start to add 500 people every other month to learn what we're doing in Parkinson's disease, it will enhance our ability to recruit volunteers for our clinical trials. It'll be able to enhance our ability to get the drugs approved. We're also trying different days and different times, as you can see. We're trying to learn which way is the best time for you, the best day. Please do not hesitate to give us your opinion. Lastly, and very important, this outreach program has two parts. One is to introduce to the people what we believe is the biggest medical breakthrough of the last 100 years, therapeutic angiogenesis. When I say introduce it, the hypothesis that Dr. Jacobs mentioned, that vascular disruption leads to Parkinson's disease, leads to Alzheimer's, leads to many horrible disease, even chronic depression, which is reported to affect 20 million Americans. If we're correct, we have to introduce that hypothesis to the neurology community, the medical community, and the medical world. We have to provide evidence for that. And then the second part is we need to find people to help us accomplish the task. Zhittya is not a big pharma company. And since we're not a dinosaur, we better be very fast and nimble mammals if we're going to succeed. Our outreach program has reached many people, and things have come in from the unknown. A world-class professor at a major university presented to Dr. Jacobs and I a way to get FGF1 into the brain other than infusions. He was part of the people we touched, and he came to us with an idea that we think may be outstandingly beneficial to all. Secondly, a world-class company in Europe with a diagnostic imaging technology to better diagnose and measure our success in vascular in the brain came to us. Once again, he was touched by this outreach campaign. A medical device group out of Berlin came to us with a revolutionary catheter to introduce our drug non-surgically into the heart. Now, before I go on, I want to make one comment. Jack and I stumbled onto doing clinical trials in Mexico. And when the people there showed us that we could get a 1-year jump on finding out if Parkinson's disease in humans can be safe and can have beneficial outcome, it made sense for us to try. Secondly, there's nobody in my path of life had ever told me that Estonia, once part of the Soviet Union, had no drug approval process. It all came from Moscow. When they became independent, they developed a slight drug approval process. Then they became part of the European Union. They're an amazingly unique unicorn that may help us not only do the clinical trials, but get approval very quickly. Dr. Jacobs and I had not thought of any of this. This all came from people like you who came to us with ideas, understanding we have a huge task ahead of us. We need friends. We need ideas. And there's something else you need to understand. Neither Jack or I or anybody on the Zhittya team is fighting for our academic pride, or we thought of it first here. We have a duty to those people suffering with Parkinson's, those people suffering with diabetic foot ulcers, the people who have no option heart conditions. We believe this works, and we have a duty to succeed. We eat our pride if it means victory. If you come to us with something that will show us how to trim 3 months off getting our Parkinson's medicine to people and it working, we will eat our pride. Victory is what we're after. I was raised by a father whose definition of finishing a race in second was, you lost. We don't plan on losing this race, people. So please do not hesitate to email us with good ideas. We're open. We have a huge task ahead of us. We look forward to your help. We hope you've enjoyed this. Hopefully in a couple of weeks, we will have the presentation on YouTube. So those of you that came in at the end or the beginning or halfway can pick it up, or if you want to refer it to friends. We thank you for your time. This is Dan Montano saying good night. God bless you all. Bye.